All right, guys, I'm going to make this super quick. This is our very short lecture on endosymbiosis. So after having talked about it in class and watched the video as an intro, here are the actual notes that might help this solidify in your head. First, we're going to start with this picture, because this is the woman that developed the theory of endosymbiosis. Her name is Lynn Margulies, and she is a brilliant woman. She is no longer with us. She passed away in 2011. But her contributions to biological evolution by developing this theory are so significant. And she developed it in a time when women's ideas were not always accepted. And so the first paper that she actually wrote on this theory was rejected from like 15 different scientific publications. And she was kind of laughed at for her thoughts and ideas. However, come to find out, she is incredibly brilliant, and this idea of endosymbiosis has been supported by multiple lines of evidence that we will see here in just a couple of moments. Okay, so kudos to Lynn for sticking with it. Okay, so in your notes, you have this picture right here that is the most simplistic diagram of endosymbiosis that I could find. So it actually steps you through 1, 2, 3, 4, 5 steps. You start with 2 bacterial cells that are independently living. One bacterial cell engulfs the other, and now they exist in symbiosis. So they're hanging out together, and then as they replicate, as this bacterial cell replicates, that internal structure now is replicated in every cell that comes from that original parent cell. And so our task is making sure we know what it is and then what evidence supports it. So endosymbiosis is the theory for how eukaryotic cells evolved from simple prokaryotes that existed on early Earth's environment. So it is also the idea, or embedded within the theory is the idea, that one prokaryote engulfs or eats, right? Endo bringing into, right? We've learned endocytosis. So here, this is a very similar word, endosymbiosis, where a prokaryote engulfs or eats another, and then they live together in a symbiotic relationship. Symbiosis, remember, harmony, nobody's attacking the other, and usually that's because both organisms are doing something that benefits the other. And so they kind of tolerate or put up with each other because they're getting something from each other. So the outer prokaryote, the one that engulfed, the one that did the eating, is now receiving an energy strategy, a metabolism strategy from the inner prokaryote. And the inner, the one that got eaten, is receiving a bit of shelter and protection from the outer. And so there is the symbiotic relationship between the two of them.
Surface formation.
Here is a nice little video that just lets you watch the modeling of that process. So take a look at this. Get back there. And I'll just let it play right here.
Surface formation. These cells were prokaryotes, cells lacking organelles or other internal membrane-bound structures. It wasn't until 2.7 billion years ago that eukaryotes, cells with a nucleus enclosed in membranes, appeared in the fossil record. Eukaryotes evolved from prokaryotes. Here is how that is thought to have happened. A prokaryote grew in size, and as it did, its surface area to volume ratio decreased. This is a problem because it now needs to feed more bulk, but it has less surface area to gather nutrients. So, to increase the ratio again, the cell developed infoldings in its membrane. Eventually, these infoldings pinched off from the cell membrane to form an early endomembrane system surrounding the nucleoid. This was the first membrane-bound nucleus, and hence this was the first eukaryotic cell. This eukaryotic cell endophagocytosed an aerobic or oxygen-using prokaryotic cell, which may have been prey or a parasite. Genomic sequencing of cells today indicates that this prokaryote was from a group of bacteria called the alpha proteobacteria. In any case, this cell avoids digestion and becomes an endosymbiont. In other words, a cell living within another cell. Funnily enough, this endosymbiont became useful to the eukaryote. The aerobic prokaryote was able to use oxygen to make energy, a process called respiration, which was nice because the Earth's oxygen concentrations were increasing at this time due to the activity of cyanobacteria. The aerobic prokaryote benefited from its host because the cytoplasm was full of half-digested food molecules. Digesting these molecules with oxygen, the prokaryote produced so much energy that some of the ATP leaked into the cell's cytoplasm. Hooray! So, as other eukaryotes went extinct from the rising oxygen levels, the eukaryote and the endosymbiont became best pals, with the endosymbiont becoming a mitochondrion. At this point, the endosymbiont became an obligate endosymbiont, meaning it cannot survive on its own outside the cell. Sometime later, the same process occurred with the cyanobacterium, which became the chloroplast.
Okay, so I know there were some bigger words in that video. Hold to just what we were able to visually see in this model, and also the idea that one cell engulfed another and they lived in a symbiotic relationship. Okay, so digging a little bit more into that, here is a timeline of pictures, right? So the aerobic prokaryote that got engulfed, we think became eventually present day mitochondria. That is the organelle that does what the aerobic prokaryote did, right? It uses oxygen to create ATP. And the photosynthetic prokaryote that got engulfed eventually over time became this internal structure of the chloroplast, because this is the organelle that does what this photosynthetic prokaryote used to do, use the sunlight to create an energy source. So when we look at evidence that supports endosymbiosis, we're always going to look at the chloroplast and the mitochondria and pull evidence from those two present day organelles. So we look at chloroplast because we think that they most likely were a photosynthetic bacteria that got engulfed. And we look at mitochondria today because we most likely think that they were an aerobic bacteria that got engulfed in the process of endosymbiosis. So when we look at evidence, there's three primary lines of evidence to use and support. So these next 3 points of evidence basically try to show how the chloroplast and the mitochondria as organelles today resemble prokaryotes. How could they possibly have been prokaryotes? Where's the evidence that supports that a long time ago the chloroplasts and the mitochondria were actually free living prokaryotes in the environment? Well, the first relates to the membranes. So the first line of evidence goes back to the idea that the chloroplasts and the mitochondria are double membrane bound organelles. And so here, the inner membrane, so the folded up inner membrane of the mitochondria, the inner membrane of the chloroplast, that was most likely the prokaryote's original membrane. And then through the engulfing process, look up here at this diagram, endocytosis, remember, put something into a vesicle, and the vesicle came from the cell's membrane. Well, now we have the outer membrane, which really is like the vesicle that the cell was surrounded in initially when it was taken in. So the fact that there are double membranes, that points back to, hey, a long time ago, the inner membrane was the original prokaryote's membrane. And then through the process of endosymbiosis, when it got put into a vesicle and brought in, it created this outer membrane, giving us two. The second line of evidence has to do with DNA. So we know that a cell has DNA inside of its nucleus. That's called nuclear DNA. However, really cool thing, mitochondria and chloroplasts have their own DNA that's completely separate from the nucleus DNA, the DNA that's hanging out in the nucleus of a cell. And that's because we think a long time ago, the mitochondria and the chloroplast used to be their own independent cells living freely. They had their own DNA. Also, if you caught in the video from class this week, not only do they have their own DNA, but their DNA is in a circle structure, which is exactly like the DNA in prokaryotes today. They have one circular chromosome. And then the third is looking at reproduction. So both chloroplasts and mitochondria can reproduce on their own, meaning the overall cell doesn't have to be going through mitosis for these organelles to duplicate. All the other organelles, the cell has to actively be engaged in its cell cycle, getting ready for mitosis for those organelles to duplicate. These guys, the chloroplast and the mitochondria, duplicate on their own, which again comes back to the idea that they used to be free living cells that had to duplicate on their own whenever they needed to. And not only do they duplicate, the duplication process looks a lot like binary fission, which is the grow and split yourself in half process that bacterial cells do today. So, long story short, I'm summing this up. What is endosymbiosis? The theory that suggests that we now have complex eukaryotic life with all of these internal organelles because one prokaryote engulfed another. What do we look at to support this theory? We look at the organelles present day that we think a long time ago were those original cells that got engulfed. We're going to look at chloroplasts and mitochondria. Why do we think chloroplasts and mitochondria a long time ago were free living prokaryotes that got engulfed? Because of all these reasons that we have cited right here. All right, I said that I would keep it short, so I'm not going to elaborate. That's the end of the lecture. I will see you all on Wednesday.